Okay, I think I'll just go ahead and get started with some of the preliminaries. I'm Charlie Nunn. I'm at Duke University in evolutionary anthropology in the Duke Global Health Institute, and I also run TRISEM, the Triangle Center for Evolutionary Medicine. TRISEM organizes these Club of Meds with a bunch of other evolutionary medicine centers, and I'm going to paste that into the chat. Hopefully, did that go into the chat? Okay, I can't see it for some reason, but anyhow, there are a whole bunch of other centers out there that help us organize this, and I want to thank them and recognize them. And I also want to thank the International Society for Evolution Medicine and Public Health and our journal, um, which are also involved. We often try to highlight articles from the journal, of course. And I just wanted to point out that there are a couple of new special issues. Well, there's one new special issue, one existing special issue. There's a new one on microbial evolution in human health. Looks like a fantastic special issue that's being organized, I think, by Robert Woods. And so please check that out. I put a link in the chat. And then there's also an existing one that's been open and I think is closing this summer, run by my colleague Jason Somarelli on Evo applications in cancer. So please consider those opportunities to publish at EMPH, Evolution Medicine and Public Health. And I also want to point out that the conference registration is now open for ISEMF, International Society for Evolution Medicine and Public Health. The conference will be in Kiel, Northern Germany, beautiful place, July 28th to the 31st. And there's a link in the chat for you to register for that conference. We've got a great lineup, a lot of good plenary speakers already lined up. Okay, I think let's go ahead and get started with the speaker. It's a real pleasure to introduce Robert Dudley as our speaker today. I've known Robert for quite a long time, I think upwards of 20 years now. He's a professor at UC Berkeley in the Department of Integrative Biology. He was a Duke undergraduate and went on to get his PhD at University of Cambridge. He runs the Animal Flight Laboratory at UC Berkeley. And if you look at his website, it says we work on the biomechanics, energetics, and evolution of animal flight, of flight. It says here, I wrote it down wrong, I think, of flight. And if you look across the tabs, you'll see things like lab members, research, all the usual things. And then you see this tab that says the drunken monkey. And if you click on that tab, you're going to see a huge body of literature that he's contributed to involving this hypothesis that he has been the proponent of and really developed and brought real scientific rigor to with all of the work that he's done on this topic. And as I said, I've known Robert for at least 25 or for about 25 years, and he's been working on this topic for at least that long. Now, we thought that with dry January, it would be a good opportunity to highlight this hypothesis and the perspectives that Robert has been developing over these years, and to just hear his take on why humans have such a propensity to consume alcohol. So I'll turn things over to you, Robert. Thanks for joining us today.
Okay, great. Thank you, Charlie, for the introduction, and thanks for the opportunity to speak to this audience. I do work mostly on flight biomechanics, so it's a little different from the theme I'm going to cover today, and I'm actually going to cover three separate themes. One of which is primatology. I'm not a primatologist, okay? Another theme is alcohol consumption by modern humans. I drink from time to time, as do many of us, but I'm not a clinician. I will talk about alcoholism. And I'm not really an ecologist. I work in biomechanics, but I spend a lot of time in tropical environments. So I do see a lot of primates and other animals eating fruit and nectar. So these are three distinct fields for which I have no particular expertise, but I want to try to convince you that at the intersection of these three fields lies something I call ethnology, the comparative biology of ethanol consumption in natural environments. So this is the drunken monkey hypothesis. I first started working on this about 25 years ago, and the ball is still rolling, as we shall see. Can we link modern day human drinking patterns, as exhibit A in the lower right, to ancestral biases associating ethanol consumption with nutritional reward, as possibly manifested by this chimpanzee on the left? So where do we start? Let me just try. Okay, so The first paper on this topic I wrote unabashedly speculative in year 2000. Year 2014, I published a book called The Drunken Monkey: Why We Drink and Abuse Alcohol. So I've had decades now to read extensively in primatology, and I've loved every moment of it, and it's just fantastic. What was I thinking as an undergraduate at Duke? I should have been in the anthro department. Anyway, it's better late than never. And the more I read, the more I became convinced that so many of the features of human sensory and behavioral biology are linked with those not just of course, the chimpanzees are nearest closest living relatives, but also all the deep time primates. And of course, many features go back way beyond the primates. But I started to see that my own gestures and gesticulations were variably simian in character, and even the way I was just walking around, and then how was I thinking about food, or what about interacting with conspecifics or interacting with my dog? Was this all just part of, I'm on autopilot most of the time from this. So it's certainly possible. This is Alan Houle's picture in Kibale of a fruit of a chimp smelling a cracked open fruit. And I just thought, wow, look at the bouquet. Look at the facial gestures, their fingernails, look at the ear structures. We're not that far removed, although we like to think that we are. So I'm having trouble advancing the slide here. Hang on. Okay. When I wrote the book, I first started with the drinking habits of Charles Darwin. I thought it was kind of fun. No one worked on this before. And you can sort of go through the literature and pick up a lot of stuff. He sometimes drank to excess as an undergrad at Cambridge. Moderate consumption, a glass of wine daily at Down House. He was very sick in the latter half of his life, so he was regularly prescribed the brandy and wine, but he was really careful. And one of the reasons he was careful is that there was a family history of alcoholism. So he was kind of wary about this stuff, but this was just kind of a fun thing to do. More importantly, in a unpublished letter, now it's published, the Darwin Project has got everything, I believe, now online. This was a letter, and of course, he was a voluminous correspondent all over the world. How did he find the time? And he wrote all those goddamn books too. Impressive. So most monkeys would be habitually attracted to alcohol if they could get it. Now, he didn't draw the link with ancestral diet. Had he done so, we wouldn't be here today talking about it. But there was a tradition in the 19th century of Europeans rampaging around the planet would often bring home juvenile primates. They were shipboard mascots. They were given to publicans, from whence derives the word pub, and basically would be fed alcohol by the humans, and they were playmates, and they were always getting into booze. So if they could get it, and they were getting it from humans in this particular context, but it was well known in the 19th century that their primates did like to drink if they could get the booze from the humans. So another giant, of course, of the 19th century was Pasteur, who worked on fermentation with ample support from the French wine industry. And then he figured out where does this stuff come from anyway? It was a mystery for a long time. So it's a biotic effect. Fermenting yeast anaerobically metabolize sugar to ethanol. Even in high oxygen concentrations, they will flip to anaerobic metabolism, which yields a much less net energy yield in the short term, but it produces this particular molecule called ethanol. And we use the word alcohol colloquially, but it's really ethanol is the 90% of all fermentation products, and that's kind of what we go for as humans and have distill and all of this. So I'll just use ethanol and alcohol synonymously from now on. What's really interesting, you ask why? Why are they doing this? So yeast metabolism is inhibited by ethanol concentrations greater than 8 to 10%. Bacteria have much lower lethal thresholds. So one of the big hypotheses out there is that basically the ethanol is being produced as a microbicide by the yeast to kill off competitors within the body of a ripe fruit for access to sugars. And the origin of fermentation in yeasts are ancient. They're marine yeasts. They go way back, but the actual fermentative pathways turn up only in the Cretaceous, coincident, plus or minus 5 million years with the origin of large, sugar-rich, fleshy fruits produced by the flowering plants. So that was an interesting correlational sort of thing. So maybe they're just producing it to kill off bacterial competitors, as would be implied by this sort of difference in the yeast metabolic in the susceptibility to ethanol concentrations. This is why you can only homebrew up to 5 to 10%. It's really hard to get higher concentrations, but we still use ethanol or propanol now as a microbicide as a clinical swab prior to giving injections. It just kills bacteria. So I first got into this. I like, great, what's the ethanol content out there? And why was I thinking about this at all? Well, I had the great opportunity as a postdoctoral fellow to live on Barrow, Colorado Island for 5 years, not 5 months, that's 5 years. And it was just a wonderful opportunity to live in the tropics. And I was working on insects, but the primates were everywhere. I'll talk about that later when we have questions. Here's Barrow, Colorado Island, magnificent forest, oldest studied tropical forest in the world. It's just this amazing database, field access, primates, butterflies, toucans, you name it. It's all there, and a lot of fruit. So I wanted to work on fruit. So that's what's the ethanol content of a fruit that's consumed by a primate. And, most of the primatologists know this well. Of course, palm and fig fruits are keystone resources, are consumed not just by a lot of primates, but by a lot of animals, birds, and mammals. So I thought, all right, let's just look at a palm fruit. And conveniently, this is Astracarium stanleyanum. That's about a meter-long infructescence, probably weighs 50 kilo. It's beautiful fruit. It's delicious. Okay. So you can, and lots of animals go on to this. I thought, well, let's just measure ethanol content within these fruits. Now, you might think, well, the tropical forest, they got a lot of fruit. Actually, fruit's very scarce. It's episodic. It's hard to find, particularly looking through a vegetational canopy. If we are dropped down in the Amazon, we random walk for about 2 or 3 days before we run out of food and starve to death. It's hard to access ripe fruit. So we think there's been strong selective capacity for the ability to localize and get to fruit quickly when you can find it in the forest. And I'll talk a little bit more about that later on. So here's the spider monkey in Panama going after the fruit. We don't know how it found this infructescence, but they tend to binge feed, they scrape, they throw the fruit, they scrape again. So there's basically, there are a lot of things that eat this fruit. This is widespread palm and lowland neotropical forest. So here's just not a complete list of everything on BCI, Barrow, Colorado, that's actually eating this. And I thought, great, a couple primates, we know it's been studied for other kinds of reasons. And they're only eating the pulp, they're discarding the seed. So we thought, okay, let's just do this. And if there are any fly biologists in the audience, this is what happens when a scraped fruit is gratuitously thrown to the forest floor, and then a wide range of fruit flies immediately come to it and mate an oviposit, and within 2 or 3 days, affect the next generation. So the biology of fruit flies is actually also dependent, not all fruit flies, but a lot of them dependent on the ability to find these fruit remnants and then carry out a full life cycle on them in the forest. If anybody's had the experience of you're outside and you crack open a beer bottle or open a bottle of wine and all of a sudden there's a fruit fly on that bottle. How did it get there so quickly? How did it find the fruit? What was it doing before it found the fruit? You don't really see them unless there's some sort of nutritional thing for them to come to. So fruit is interesting. Ripeness just indicates suitability for consumption via vertebrate dispersal agent in this mutualistic interaction between vertebrate dispersers and the flowering plants. We can measure sugar. That's an easy one. Primatologists do this a lot. Ethanol is a lot harder. We can talk for hours about the difficulties of doing this under field conditions. First modified breathalyzer is portable gas chromatograph, infrared spectrometers now, and some other chemical test strips. Texture, fruits before the seeds are ripe are trying to defend themselves from being consumed because you lose the seed. So they tend to be hard and green, and then they soften up, and the complex carbohydrates are converted to simple sugars that are then suitable for consumption. You can measure the texture. Color is an easy one for spectroradiometer. What nobody's done for wild fruit is yeast and bacteria population densities. Going back to Pasteur, if the yeasts are producing ethanol to kill off bacterial populations, we should see an interesting time course of the relative sizes of these two competitors through time. So it's a time series and it's hard to measure, but that's an interesting target downstream. So here are the fruit, which you get by throwing stones or branches at trees. It was a wonderful skill to develop. A refractometer, spectroradiometer, modified breathalyzer is used by your local friendly police department, and then a texture meter. They've got better this is 20 years ago, so we got better iterations. And just to remind you, the fruits, again, they start out green. This is reflectance versus wavelength. They turn ripe and then they go overripe and ultimately rot. Now, what does this actually mean? These are my a priori anthropogenic categorizations of what the fruits look like to me, but that's not necessarily what the primates are eating, but I just did this ahead of time. So how much ethanol is in the pulp? The animals are eating the pulp, discarding the seed in most cases. So this is the first result in addiction. Ethanol is a function of sugar concentration. Unripe fruits have no measurable ethanol. There's just nothing there because there's no sugars. They're all complex carbohydrates. Some ripe fruits, using my a priori characterization, have zero ethanol, but obviously this is a complex sort of or microbial phenomenon whereby it either takes off or the initial inoculation is different, and so on. Unripe fruits, ripe fruits have measurable ethanol in some cases, and the overripe ones have a lot of ethanol, 8%. This is an ethanol vapor pressure inference. So, there's ethanol out there. I think that was the take-home message, and it could be really high. And you don't need a breathalyzer or GC to realize that these fruits taste really good, and they do taste alcoholic. So this is a first proof of actual presence of the compound in a natural wild fruit consumed by primates. So let's hypothesize. You could use the ethanol plume to localize a ripe fruit crop. It's a very volatile, low molecular weight, volatile molecule, so it goes great distances. If you can smell it and fly upwind or move upwind, you can get to the fruit crop quickly. More importantly, you get there before others get there, and then you can binge feed and consume as quickly as possible. So maybe there's preference for ethanol containing fruits, concentration dependent selectivity. Ethanol obligately derives from the fermentation of simple sugars. So where there is ethanol, there has to be sugar. This is a primary constituent. All the lipids are involved sometimes, but also this is the main thing that the frugivores are going for, is the sugars, at least at the macronutrient level. And then maybe ethanol consumption via frugivory is associated with caloric gain and ultimately with satiation. They get there, they find it via ethanol cues, they feed psychoactive hedonic reward, they consume, but if the ethanol concentrations are low, they're going to be consuming a lot of fruit pulp, and then their bellies should fill before they reach inebriating loads. That was another idea that we had. That's why you don't see drunk animals very often, because their stomachs are filling up before they can reach the concentrations we can reach in our bloodstream in just a minute or two via distilled products. So these are just ideas kicking around 20 years ago. Let's ground it in reality. Let's look at flies. Drosophila and ethanol. What do we know? They follow ethanol plumes to find the oviposition sites. This has been proven in laboratory contexts. There's genetic variation in the ability to metabolize ethanol. Again, from a Darwinian perspective, only heritable variation is going to provide the substrate for selection on particular traits. This goes back to the 60s. There are latitudinal gradients in ethanol availability in fermenting fruit. Or so it was hypothesized, and the alcohol dehydrogenase and aldehyde dehydrogenase variants kind of broadly track that worldwide. It's a very coarse analysis, but that's out there in the fly literature. But here's the point that I really want to make. Both lifespan and fecundity of Drosophila increase at intermediate levels of ethanol exposure. That's crazy. This is called nonlinear dosage response curve, or hormesis. In toxicology, how can this be? How can ethanol ever have some influence on fruit fly lifespan? Here are some data collected by others for totally unrelated reasons. You grow up flies across the lifespan in ethanol vapor of different concentrations, and you get this massive reduction in mortality at low concentrations of ethanol exposure relative to either zero or high levels of exposure. What is going on here? They don't know. It's unclear, but it may get back to the antimicrobial activity of the ethanol molecule. That's a hypothesis. And maybe it's killing off infections inside the fly body. And we're going to talk about comparable curves for humans in a little bit later. So let's just think about comparative biology of ethanol ingestion. There are lots of anecdotal accounts of drunken animals. I collect them. Send them to me, please. But they're not scientific. They're not grounded in measurement. They're just, if you see an animal behaving strangely, it could be due to toxins or disease, we really don't know. There are only two cases of actual liver toxicological screens in apparently drunken animals, cedar waxwings. And it was death by ethanol feeding on fermented fruit in the spring thaw. That's the only real data. And again, the argument is they're maybe not getting ethanol to high levels because their bellies are filling up and they just can't push the fruit through quickly enough to get to the high level. So we've never measured blood ethanol levels on free-ranging frugivores. We know a lot about modern humans, but the research project would be chimps on breathalyzers at this stage, and I haven't gotten the wherewithal to do that one. Just to give you some idea of the diversity of animals that could feed on fermented fruit, so it's not just the primates, it's basically a lot of other things. About 40% of the tropical butterfly fauna feeds not on flowers, but on fallen fruit. And so there's a lot of interaction. There's different morphology to the proboscis and other kinds of things. So this is a comparative biology of ethanol exposure. Large catfish of the Mekong, Amazon, and Congo basins feed on fruit during the rainy season. The fruits synchronize to fall into the river. There's long distance dispersal by these giant catfish. The fruit goes anoxic immediately in the water. So I don't know if there's an actual linkage with the fish, but it would lend, were there to be one, it would lend new meaning to the phrase drinking like a fish. I still think there's something interesting in this topic. And of course, the fruit bats. And there are so many radiations of fruit-eating birds and other kinds of things as well. So I thought, well, this is a kind of a broad idea. Is there ethanol in these fruits? This is the real question. I showed you some data that there is, but what about all fruits that primates are eating? So I want to talk about ancestral associations between primates and ethanol. And there's a large, let's start in China. There's a large mythological tradition between monkeys and wine, temple wine, and Journey to the West, the Monkey King is always getting into trouble and stealing wine and just wreaking havoc. So it's a wonderful anthropology of monkeys and alcohol. But here's a chimp in a tree in Uganda, and it's picking fruit. We don't know how it picked that particular tree. We don't know how it picks particular fruits and so on. But there are a bunch of different cues that they're potentially using. We can just watch it. It's a happy chimp. A lot of figs. It's not eating all of them. It's selecting some of them. Just cycle through. Let me just go back and play it once more. So it's going to touch, looking around, touch, looking around, pick up, smell, and then consume. So my point here is that there are actually multiple cues, even in the short-term selectivity that involve vision, palpation, and olfaction relative to fruit selection. So if you clandestinely watch your fellow humans in the supermarket, you will see exactly these behaviors. It's really quite interesting. The clandestine squeeze, smelling, looking around. It's, again, these are we've inherited something from these animals or from our nearest recent, from last common ancestor. So olfaction can't be ruled out at short distance, and it certainly can't be ruled out at long distance. If fruit flies can do it, then certainly you would think fruit-eating mammals could do it. So let's ask, well, what's the historical exposure potentially of the primates as a whole, and hominoids and hominids particularly, to fruit? And I'm going to make the case that actually, where there are simple sugars in the tropics, and there are yeasts, the yeasts are everywhere, they're on us, they're on all flowers and fruits, and high temperature, high humidity, there is fermentation. It's not a question of presence, absence, it's a question of relative degree, and then how much of the animal is consuming. But you go back 24 million years ago, all the way actually back to origins of primates 55 million years ago, they were frugivorous. Now, this is not my claim. This is just, I'm trying to replicate what people say in the literature who actually know what they're talking about. These are paleontological data, but mostly based on dentition and paleoecology and a lot of other things. I realize it's a much more nuanced sort of problem, but just to give you a strategic overview, all the way up to 5 million years ago in the origins of hominids, people have inferred predominantly frugivorous diets. So we can't recapture behavior, feeding behavior from the fossil records. So this is a big problem with this analysis, but there definitely was fruit and perhaps majority fruit in a lot of the tropical primates at least. Another way to look at it is ask what the nearest living ancestors are doing. Gibbons, orangs, chimps, and gorillas are all predominantly frugivorous. Again, it's nuanced, but they eat ripe fruit. As far as I know, gibbons and orangs almost exclusively eat ripe fruit. And then in the last 5 million years, the hominids are moving on to everything else. Sushi by the seashore, tubers, of course, and roots and this kind of thing. And they rudely eat one another from time to time. I think that's documented in the paleontological literature. But it's not that... Sorry? Okay. So basically, there's a whole bunch of we call this an ancestral heritage, sort of link between the hominid diet and exposure to fruits that potentially contain ethanol within tropical environments. So the best way to look at this is phylogenetically, of course. So if we look at the apes, lesser and greater apes, this is an intentionally misleading presentation. So percent frugive rate, less than 5% or greater than 50%. So they're all greater than 50%, including the reconstruction for hominin species, which would include Homo sapiens. The highland gorillas, of course, don't eat fruit. Above 2,000 meters, the energy balance is insufficient for plants to produce large sugar-rich fruits. They shift on smaller fruits, and then the gorillas up there eat many other things, but they can't eat big, squishy, fermenting fruits because there's nothing available up there. So this just suggested to me, well, maybe we've just inherited ancestral bias associating ethanol with nutritional reward. Again, it's just speculation, but seemed well grounded in at least the context. And then really want to say something about this is a quick talk. Human alcoholism. Does it tell us anything about the propensity to consume to excess? What is alcoholism? Well, the definition is labile. It varies by decade, it varies by country, incredibly variable. Churchill started drinking before breakfast and wrote 63 books over the course of his lifespan. So was he an alcoholic? How would you, depends on the definition. But what we do know, clinicians know it when they see it, narrow sense heritability estimates range from 0.2 to 0.6. It's partially heritable. If it weren't heritable, then probably there's no evolutionary underpinning to it. This is sort of well established. Here's where it gets more interesting, linking with fruit fly biology. Genetic variation in ADH and ALDH is correlated with the propensity toward abuse, both within and among populations. What do I mean by this? These are the two enzymes that are primarily degrading ethanol. In East Asia and only East Asia, so modern day China, Korea, Japan, Taiwan, just broadly generally construed, there are variants in these enzymes, the different alleles that basically render the carriers very susceptible to the inebriating and toxicological effects of ethanol. There's acetaldehyde buildup, and it causes a lot of symptoms if these individual carriers drink just small amounts of alcohol. So just to drive this point home and make this a memorable talk, I'm going to say a few words in Mandarin, and then I will translate, and you'll see the relevance. 女士们、先生们，你们好，大家好。我跟你们说，我的老婆是中国人，她真不会喝酒，白酒、啤酒、葡萄酒，她喝一个真小杯子，刚刚吹了，坏了，不好的东西。So what I just said in Mandarin is that my wife is Han Chinese from mainland China. And she has a fast-acting ADH and a slow-acting ALDH. If she drinks 2 milliliters of ethanol, it's amazing. She's just knocked out. The red face, the heart, the perspiration, the tendency to fall down. Just can't drink at all. So this is a set of traits she has in common with over 1 billion people worldwide, but of course, mostly in East Asia. Now, historically, it's always been claimed that rates of alcoholism in East Asia were much lower than those in other parts of the world, particularly Western Europe. But that's a culturally badly confounded comparison. So it's not, and my point here is actually, if you get sick when you drink alcohol, you tend not to drink alcohol at all, so you can't become an alcoholic. So they say, well, maybe there's a link with protection against alcoholism. But that's a complex situation culturally. If you look, however, within East Asian populations today, so within Taiwan and Korea, you look at people who are classified clinically as using local criteria as alcoholics. They are 10 times more likely to have the non-East Asian versions of these two enzymes. So they're much more like the West Europeans and the North Americans, as it were. So it's the only known genetic variant that is protective against alcoholism as a clinical disorder. You have to inherit this one. Now you can override it. You can drink. You get your liver to produce more of these sort of different kinds of enzymes, but it's still a very pronounced effect in East Asia, and only in East Asia, as far as it's known. The question is why, and I'll talk about that a little bit later. There's also some epidemiology suggesting that chronic low-level ethanol consumption reduces cardiovascular disease and overall mortality. And I'll get into the complexities of this in a moment. My final point here is that exposure to high concentrations of ethanol is fairly recent. So distillation only turns up less than 1,000 years ago in Central Asia, what is now part of Western China. So all this suggests an evolutionary mismatch between the low concentration, nutritionally linked consumption of ethanol via frugivory in primates and human ancestors relative to the concrete jungle that we walk into today. And it's like unlimited availability of high concentration booze. Here's just one of many meta-analyses of the relative risk of mortality versus daily number of standard drinks in modern humans. This is a meta-analysis. Every 6 months, the New York Times publishes new findings of a different meta-analysis, which either confirms or contradicts this U-shaped dosage response curve for modern humans. Should we drink more? Should we drink less? It depends on genetic background. Talk to your clinician. Depends on your sex. What standard drink is not standardized across countries, by the way. It's either 10 or 12 grams of ethanol per drink, depending on what, Western Europe or North America, this kind of thing. But this mirrors the epidemiology of the fruit flies, of course. So this is a long-standing debate. National Academy of Sciences and Medicine just did 2025 a huge meta-analysis of meta-analyses, as it were, and they concluded that this finding is actually correct, that there's a sweet spot somewhere for low-level ethanol consumption relative to either high-level consumption or abstention. And it's very complex epidemiology because, of course, it's all retrospective. It's all based on self-reported drinking data over the lifespan. How reliable is that? It excludes ethanol consumption via foods such as kombucha or yogurt or apple juice or fermented bread products or so on. So it's a huge public health question, though, because look at this massive reduction in the risk of mortality. The agent of protection is unclear. So is ethanol helping to kill off microbial populations in our mouth and throughout our body? And most of the effect derives from protection against cardiovascular risk, but a lot of arteriosclerotic plaques are initiated by bacterial infections. So I think the jury is still out. Read the New York Times, read the National Academy of Sciences and Medicine. It's a very interesting article. Most people worldwide do drink or either intentional ethanol consumption or they're getting it via kombucha or other fermented products. They're all getting exposed to ethanol. So here's to the theme of evolutionary medicine, the evolutionary context. Natural selection has acted on human ancestors to associate ethanol with nutritional gain, promoting rapid localization and consumption of ethanol-containing resources. Heritable behaviors that were once advantageous are now disadvantageous. It's the mismatch hypothesis. So too for lipids and sugar availability, all these diseases of nutritional excess, we just go haywire. We can't break out of it in modern times when we've just given this nutritional overload. So that's the hypothesis. Ooh. What's going on here? Okay, some people don't like this idea, and it was great. The major critique I got early on was that I'm not a primatologist. So what do you do with that? But these two people were friends of mine. We had a conference in vino veritas. It was great about 20 years ago. It's really a plea for more data. Wild monkeys and apes avoid consumption of overripe fruit. Ethanol concentrations in consumed fruit are low. Inebriation, if it occurs, is extremely rare. Again, we really don't have a lot of data. Doug Levy, also a friend. Furthermore, frugivores strongly prefer ripe over rotting fruits, even though the latter may contain more ethanol. Data on ethanol content of ripe and rotting wild fruit are lacking. So we need more data. And the best definition of science I ever heard is that we are advancing the frontiers of ignorance. We're just making more complex questions to force us to get more data. So this is a paper in 2008 on fermented nectar consumption by tree shrews. And this is the tree shrew and a slow loris as well, a palm in Southeast Asia. And they're lapping up fermented nectar. In the lower right, you can see the frothing white of the flower embedded in there. That's fermentation. And they're getting predictions for high levels of ethanol consumption on a chronic basis. And in the lower left, there's a marker, hair ethyl glucuronide. This is used forensically to assess drinking in modern human alcoholics. It's turning up in the animals that are consuming the fermented nectar regularly, but not in the other sympatric mammals. So this is kind of cool. This means, nectar is just sugar water, and the yeasts are in there. They're getting in there from the insects that visit the flowers or encapsulating it as the flower is developing. And they start fermenting, and flowers in tropical environments they're warmed up and it's humid. So all nectars are potentially fermenting. It's not just that. This is a great paper in 2015 by others. The origins of ethanol consumption prior to human-directed fermentation. There's a point mutation 12 million years ago amongst the great apes that lets them metabolize ethanol 20 times faster than that of their predecessors. And this is coincident approximately with terrestrialization and bipedalism in the great apes. They advanced the interesting hypothesis that they're accessing fallen fruits on the floor of the savanna or of the jungle that have had longer time to ferment. That's a greater source of ethanol for these animals. That's speculative, but this is they reconstructed the enzyme and showed that it actually has much higher catalytic efficiency. So that was a good one. We did a paper a couple of years ago on dietary ethanol ingestion by spider monkeys in Panama. Measure what's in the ethanol. We then measured from the urine samples that secondary metabolite of ethanol, and 6 of 7 of the samples turned out highly positive and significant. And then my student Alexei Maro has been working on chimpanzees in the wild to see what are the ethanol contents of the majority fruit species that they consume through the year. So I have data for a lot of different species, I think about 25 or 30 maybe, which represent about 80% of the diet at 2 different field sites. We measured ethanol content using 4 different methods. And the global average is 14 grams of ethanol per day. This is the paper in Science Advances, which now miraculously has been, just last fall, it's been downloaded more than 17,000 times. I couldn't believe it because it was just, for me, it's indirect corroboration of the drunken monkey hypothesis. We have this obsession with alcohol. Everybody wanted to read about the chimps as our nearest living relatives. That, wow, they're getting 14 grams of ethanol a day, and they're about 60% of our body mass, all things else being equal. So that's about 2 standard drinks a day, which sort of matches the inferred sweet spot based on the epidemiology. So, and we've also done recent work on floral nectar in the upper part of the graph. Ethanol is weight by weight. 0.02% doesn't sound like much, but hummingbirds and honeybees and things like that are drinking 50 to 80% of their body weight a day in nectar. So this is measurements done in the UC Botanical Garden, which is not a tropical environment. It's cold, temperate, Mediterranean type climate, but there's ethanol in flowers. And depending on how much these animals are drinking, they can also get substantial loads. We calculated that for hummingbirds. So ethanol is widespread in fruits and in nectars. It's just a question of relative extent and how much are the animals actually consuming. Almost done here. So here's a nice paper on that. One of the polymorphisms in East Asia, you can see this is a clinal distribution of this slow, fast-acting alcohol dehydrogenase. And it's basically centered around modern-day Shanghai. And as you go out in any direction, basically the frequency diminishes. The marks and the various stars and stuff are historical sites of crop domestication over the last 12,000 years. So and they dated, by the way, this polymorphism to about 10,000 years ago using molecular clock. So it looks as if this very interesting sort of genetic variant only found in East Asia is at least temporally coupled to the origins of crop domestication. That would be rice, sorghum, and millet in the 2 great river valleys, the Changjiang and the Huanghe. So, what's the linkage? They don't know. Nobody knows. It's thought it might be a third participant, that somehow these polymorphisms are rendering the individuals more resistant to hepatitis C, which is also endemic in approximately the same area. But it's one of the great questions to me, I guess, as a non-practitioner of human evolutionary genetics, like this is such a pronounced variant relative to the rest of the world. It's very recent, and it links with domestication and presumably civilization. It could be other things. Anyway, this is why a lot of East Asians have the facial flush and the problems with drinking alcohol and tend not to drink much or any alcohol. So let me end with a series of questions. How do frugivores find fruit over long distances? That's hard to answer in the wild. We've got some ideas. What are ethanol concentrations within fruit, and what are blood ethanol levels in animals that are consuming the fruit? Hard to answer. We've got some ideas. Does ethanol act as a feeding stimulant, the aperitif effect? It does in modern humans. That's been proven by behavioral psychologists. But what about other animals? If there's a scramble competition to consume resources in the field, maybe it's just like all these hedonic rewards and socialization effects are basically just helping us feed faster and better, and so that others don't steal our dinner. what are the hormetic effects of ethanol on animal frugivores? So the nonlinear dosage response curve has been corroborated in fruit flies and also in lab rats and mice, two separate studies, but never in any other taxon. We just don't know. If it's a widespread compound, we ought to see it having broad scale sort of lifespan effects deriving from chronic exposure. My favorite quote of all time, after I read about the dangers of drinking, I stopped reading, attributed to Henny Youngman, the great Borscht Belt comedian. And I thought, this is just so cool. It's a qualitative expression of the nonlinear dosage response curve. There are costs, but there are also benefits. Where is the sweet spot? We just don't know. The majority of the world's population is probably consuming alcohol, either intentionally or unintentionally via fermented food products every day. So to finish with Velázquez Los Borrachos, the divine entity giving the gift of fermentation to the mere mortals. It's an incredible molecule. It is widespread, but it can either inspire us or it can destroy us. So to really understand why, we have to have an evolutionary perspective. I'll stop there and answer any questions. Thank you.
Thank you, Robert. That was fantastic. Really interesting, really fantastic work. We have a few questions in the chat. I wanted to start just with a quick question on one of your last slides there on that alcohol dehydrogenase variant.
Sure.
Did they also look for evidence of selection on that variant?
Not as far as I know. I think they just try to reconstruct the timeline, and there's been no follow-up on that, either for the ADH or for the ALDH variant.
Okay.
But that would be an obvious target, right? Yeah.
Okay. And maybe just going back into the chat, there's some questions in the chat. You probably haven't had a chance to see those.
Sure.
Cynthia Bell wrote several comments actually, but the first one she wrote, I think it was the first. Some studies note this is in relation to that J-shaped curve
Yeah.
on, I think it was mortality and number of drinks, I guess, per day. Some studies note that people reporting 0 drinks a day include recovered alcoholics. I also added in there, I think some of those studies also report that in some of those initial studies, at least, those were people who were abstaining because of other health reasons.
Absolutely.
And then
Absolutely.
I think there's a third factor as well, which is, if I remember correctly, people have said that those who drink 1 or 2 or 3 drinks a day also tend to have uh, more exercise, eat a better diet, so there are like some really correlated effects there. So in light of all that, I mean, where do you think things stand? Did the National Academy of Sciences report, for example, consider all of that? Or, how do
They
we
did.
disentangle all of this?
That paper, it's a monograph. I mean, it's like 350
Yeah.
pages long. They tried to look at all this. All those issues and other confounding factors have been raised, and it's a big problem with humans. You can't do the prospective study. So what do they try to tease that apart, and they have very strict sort of data criteria for consideration of particular studies and so on. So I would just encourage everyone to look at that. Again, I'm not an expert in this field. I don't know. I'm just trying to report what other people claim. The New York Times loves the stories. It's like drinking more is good, drinking more is bad. What are we going to do? Have another drink. It's like, so but it's a big public health issue. If you get these protection against cardiovascular risk, how do you balance that against all the adverse consequences of ethanol consumption?
Yeah, it's a very important question,
Yeah.
public
Yeah.
health wise. Okay, we have a lot of questions in the chat, and I think it's best if people are just willing to raise their hands or just unmute yourself and ask your question. And I'll give people a chance to do that, and then maybe I'll start calling on people. Joseph Stanford, I saw you had a question. Joe Alcock.
Yeah, I just wondered if there are studies in lab settings with animals. Other studies looking at different levels of alcohol exposure in the lab setting.
There are 2 studies that I'm aware of, one in lab mice, one in lab rodents, over the lifespan, showing that exact same U-shaped dosage response curve for, reasonable levels, not 50% ethanol, of course,
Right.
like 0 to 10% ethanol.
Yeah, but I got to tell you, I wouldn't take much stock of studies like that. 1, because they're small, and 2, because the strains they use are always inbred. So it may be true for
Yeah.
those 2 inbred strains that you get that response, but those kinds of studies are just.
I I agree entirely. And I made the argument in my book, the lab rodents are exactly the wrong model to use because they're temperate zone granivores and omnivores. There are all these great tropical rodents that feed almost exclusively on fallen fruit, fermenting fruit, that have a natural biology of exposure to alcohol. So that's what I would recommend. They don't like to hear that because they have their own inbred strains to work with. Yeah.
Okay. Joe, did you want to ask a question?
Yeah, I mean, my
Okay.
thing was that,
Other Joe.
the other Joe Alcott. Okay.
No, go ahead, Joe. Go
All
ahead,
right.
Joe.
Well, I'm just going to say, I mean, the issue on the evolution of alcoholism resistance really resides on whether there's any kind of data showing that alcoholics have less successful reproduction, not how long they live.
Yeah.
It's fitness. Things
Right.
evolve on fitness, not just lifespan. I can tell you that both of my grandfathers drank like fish and had family sizes of 12 and 8.
Right.
So
Right. No, so it is.
I don't know that that data exists.
I don't know either, to be honest. Yeah, I agree with you. Yeah.
Right. I'll jump in. So my question, and great talk. This is just a wonderful
Thank
treatment
you.
of this complicated and interesting and important idea. But my thought was that you might think that children would benefit from the antimicrobial effects of alcohol since they have kind of a high burden of GI disease and mortality, especially early in life. And yet young kids in particular show strong aversions to alcohol. So I know Ed Hagen's written a little bit about this topic, but I was wondering what you thought.
I think that the concentrations of alcohol to which children are exposed in a domestic setting are actually much higher than those than, say, juvenile chimpanzees would be exposed to in the rainforest setting. So I think instead of 5%, we should be giving them 0.5% and then see if there's aversion or, liking of the compounds. I just think we're so far removed in terms of concentrations. And also just the nature of fermentations. We only use one species of yeast. We don't use complex microbial faunas to make our fermentations, that it's just different. It's like most kids probably wouldn't smoke a cigarette either. You just gave it to them. So what does that tell you? It's just modern humans are different. I like to cut this analysis off at about 10,000 years ago, before origins of civilization and all of the cultural confounds of diet and other sorts of things. But I take your point.
Terrific. Thanks.
Okay, others who've posted in the chat or any other questions out there? Robert, you had a point about blue zones, places with high concentration of centenarians, I guess. Is that what that is?
Yes, that's
Did you
right.
wanna-
One of the things that was discussed from the original Blue Zones data was that people who are centenarians in the 5 original Blue Zones drink socially, and it was put into the category of relaxation more than anything else. And so it was kind of that was where they put it, and it was a common thread.
So, most primates are social animals, ourselves included. And if you watch dinner parties in restaurants, you'll see that the wine and the food and the socialization just all go hand in hand and presumably are beneficial most of the time. So again, that's sort of modern human cultural evolution, and also sociality is a big confound. Again, one of my points I want to make here is that this is not just a social primate argument. This is a frugivore and nectar-ivore argument, which includes vertebrates and invertebrates. So actually, there's a broad range of animal systems to which this may pertain, and it actually could be very useful for sensory and behavioral sort of aspects. But yeah.
Okay, we have a question from Tom Strusaker. Go ahead, Tom. Good to see you here.
Hi, yeah. You talk about the drunken monkey. What would be the first signs of drunkenness in a monkey?
Yeah, and how would
Have
we
you ever
know?
observed it?
Right. Exactly. So it's really kind of a misnomer. There's no such thing as bad publicity, and that sort of phrase just turned up early on. So most of the time, animals are not getting drunk. There are very few cases, proven cases of actual inebriation in the wild for monkeys or any other animal, as distinct from modern humans. So and then of course I'm talking about humans, but they're apes, not monkeys. So that was another misattribution in the title, but it's an effective title. I hope you agree at least on that. But I think, the gold standard is blood ethanol content, and then compare that to modern human information. We know a lot more about humans on highways driving vehicles than we do about animals in the wild. So these are all empirical questions. These are easily addressed if you can have access to primates or other fruit, all the fruit-eating birds and things like that, and hummingbirds too. We've been doing work on the fermented nectars that hummingbirds are consuming.
I guess I would think that selection would act against drunkenness in a species that is potential prey to a number of predators.
And particularly for flying animals. And that really is another argument for effective metabolic degradation of ethanol. So it could serve multiple functions. You use it to find the material, but then you also degrade it quickly.
Yeah, right.
Arboreality might not be a good idea either.
Stay out of the trees if you're drunk. Exactly.
And if you got chimps around, you want to stay off the ground.
Lots of possible things.
Thanks for your question, Tom. Good to see you.
Good to see you.
Noel, I think you had your hand up.
Yes. Just to answer, excuse me, Tom's question about the effects of drunkenness in monkeys, Roberta Palmer, who worked on the St. Kitts vervets, has done a lot of work on the monkeys there that are semi-wild, but they seem to be adapted to eating the fermented results of sugarcane in the sugarcane fields. So there is some observational evidence on that. But I thought your point was very interesting about the antimicrobial effects of ethanol. And I would suggest that there are some layers of evolutionary time that might be instructive in looking at this, because that would seem to be a very early evolutionary adaptation of yeast, perhaps, that then gets used in various other ways, and particularly looking at what probably happened at about 5 million years ago or thereabouts with hominin evolution, because as those populations moved away from forests, they would have moved away from fruit sources, but they would have retained possibly a taste for alcohol from the standpoint of what it was used for earlier, or we presume was used for earlier evolutionarily. And that wasn't lost, but then it was used by evolution in various ways, such as a pre-adaptation or ex-adaptation that would be related more perhaps to caloric intake. So there may be different levels of selection, which might be very interesting to look at in your perspective.
Thank you. I agree on that. Sort of the reconstruction of ancient microbial genomes is way beyond my capacity at this stage, is doing other things, but that it's potentially all available, right? So, I mean, that reconstruction of the enzymes, amongst the great apes, that point mutation, increasing catalytic efficiency of alcohol dehydrogenase, I thought was just an amazing outcome.
Yeah, exactly. I think that's fascinating.
Yeah.
Okay, I'll just note Cynthia posted what looks to be a pretty interesting article in the chat, Robert, if you want to check that out after.
Oh, okay, great.
Click on it. But Ned has a question. Go ahead, Ned.
Hi, thank you. Really great discussion and presentation. I'm going to ask a question from an obstetrical perspective, and you've mentioned the attraction to alcohol. Is there an aversion to alcohol in primates where alcohol-containing fruits are primary to their diet?
Great question, and no data that I'm aware of. So when is it aversive? So Dan Jansen, the great tropical ecologist, had a paper, I think in 1968, on why fruits rot and meat spoils or something like that. And he explicitly stated that ethanol is aversive to vertebrates. And that was sort of the dogma for a long time, that this is basically, it keeps the fruit as a fruit. And I don't quite understand the argument, but clearly it's not. It's not in humans. There's some nice behavioral work done at the Duke Primate Center by Nate Dominy and an undergraduate, showing that slow lorises and also the II prefer artificial nectar containing ethanol at progressively higher concentrations, 1 to 0, 2% to 1%, 3% to 2%. And even when they exhaust the well of availability, they keep probing as if there's something left in there that they can try to get out. But it's hard to. Those are captive animals, so there are no data on wild animals, that's for sure. And then the captive animals have kind of historically confounded diets, potentially, and other sorts of issues. So it'd be great to see more data.
Thank you.
maybe a little bit of an extension of that. I was curious about these olfactory plumes, that you talked about as a way to localize fruit. Do people vary in their olfactory sensitivity to ethanol? I feel like, some people are just they pick it up really quickly and other people can't smell it at all. And, what is the variability out there and what does that mean in the context of?
It's a good question. There are all of these other fermentation products in the bouquet, so it could be variable sensitivity. It's like insect pheromones. It's actually usually a complex blend. The fruit fly experiments are done with pure ethanol, and it's unambiguous. It's just like, wow, they're just queuing into this immediately and flying upwind and finding the fruit. So can flying birds do that? That's all the fruit-eating birds, or even partially frugivorous birds may just be sensing an ethanol plume. They're flying around all the time looking for food to eat.
That's good. Okay, well, maybe just to wrap things up, the big question, what do you think about dry January? What is your take on dry January, Robert?
I'll extend that to the absolute prohibition by the Prophet Muhammad against the consumption of alcohol, 700 CE. How do you prevent people from drinking? And the mere fact that we had to prohibit it in Arabia suggests that people were drinking it. And to this day, all the anecdotal information suggests there are a lot of end runs. Think about prohibition in the United States. How long did that last? 14 years. And there were medical exemptions, there were religious exemptions, of course, and then the bootlegging industry. Thank you, Seagrams. It was just unenforceable. So I think that's the real question: is how do you get people to, slow down on their drinking, to slow down on caloric consumption? How do you get people to exercise more? We're not selected to exercise more. We're selected to basically scavenge calories in whatever form they come. And to retain calories and to put on energy stores. So I think it's a hard sell. It might work for some people, but it's going to be difficult to really have a major impact. But I don't know. What do the data say, right? Yeah.
Okay, I feel like I'm not actually hearing that much about it this year. I don't know. Maybe it's fading a little bit, but
it's a great
I
concept.
might be reading the wrong news.
Right. How do you implement that concept? I just don't know. Yeah.
Okay, well, thank you very much, Robert. That was fantastic.
Okay.
Thanks everybody for tuning
Happy
in.
to correspond with anyone to follow up on these questions if anybody's.
I put in the chat just quickly the next two Club of Meds we have definitely on the schedule. We have a few more coming after that. And also, I want to call out EvMed Reviews, which is live. It is live in a new place for those of you who followed it. And just want to encourage people to check that out. So click on that link if you're curious. Anyhow, Robert, thank you. This was really great. And
Okay,
thanks to so many people. Good to catch up.
Thanks a lot. Likewise. See you later.
All right.
Bye.